The human gut is home to trillions of microbes, bacteria, viruses, fungi, and other organisms that together form the gut microbiome, an ecosystem essential to digestion, immune function, hormone regulation, and metabolic health. In recent years, scientists have uncovered a profound connection between the gut microbiome and obesity, with particular focus on how ultra-processed foods, UPFs, disrupt this delicate system. These disruptions don't just change what we absorb from food, they may actually influence how much fat we store, how our metabolism operates, and how our appetite is regulated. The result is a growing recognition that obesity is not just a function of calories and exercise, but also of microbial balance. The microbiome plays a direct role in how we break down and harvest energy from food. Different species of gut bacteria extract different amounts of energy from the same meal. Studies have shown that people with obesity often harbor microbial communities that are more efficient at extracting calories, particularly from processed carbohydrates and fats. In the context of a diet dominated by UPFs, which are easily broken down and rapidly absorbed, these microbes can amplify the amount of usable energy taken in, tipping the balance toward weight gain even without obvious overeating. But it's not just about energy extraction. A disrupted microbiome also influences inflammation and insulin sensitivity, both key factors in metabolic health. Processed foods tend to be low in fiber, one of the primary fuel sources for beneficial gut bacteria. Without sufficient fiber from vegetables, legumes, fruits, and whole grains, microbial diversity declines. Opportunistic or harmful bacteria, often more tolerant of high sugar and high fat environments, begin to dominate. This shift can compromise the gut barrier, leading to increased intestinal permeability, often called leaky gut. When this barrier is weakened, bacterial fragments like lipopolysaccharides, LPS, can enter the bloodstream, triggering chronic low-grade inflammation that promotes insulin resistance and fat storage. Moreover, the gut microbiome interacts with the body's endocrine system, particularly hormones that regulate hunger and fullness. A healthy microbiome supports the release of satiety hormones like GLP-1 and peptide YY, helping the brain accurately register when to stop eating. Processed food diets, by contrast, are associated with reduced production of these hormones, along with elevated ghrelin, the hunger hormone. This contributes to a biological environment where the body feels hungrier, burns fewer calories, and stores more fat, an effect mediated at least in part by microbial imbalance. Another powerful connection lies in the gut-brain axis, the biochemical signaling network linking the intestines to the central nervous system. Microbes can produce neurotransmitters like serotonin and dopamine, influencing mood, stress response, and even food preferences. A microbiome altered by UPFs may therefore promote cravings for more hyperpalatable, processed foods, creating a feedback loop that reinforces poor dietary choices and further destabilizes metabolic control. Emerging research even suggests that microbial shifts can influence epigenetic regulation, altering how genes related to metabolism and fat storage are expressed over time. This means that the long-term consequences of early life exposure to processed foods could include persistent changes in how the body regulates weight, appetite, and insulin response. These effects may be partially transmissible across generations, as maternal diet shapes the infant microbiome during pregnancy and breastfeeding. However, this complex picture also opens the door to powerful interventions. The gut microbiome is adaptable and responsive to diet. When individuals increase their intake of diverse plant-based foods, rich in fiber, polyphenol, and prebiotics, microbial diversity improves. Beneficial bacteria begin to thrive again, producing short-chain fatty acids like butyrate, which support gut barrier integrity, reduce inflammation, and improve insulin sensitivity. Even modest dietary shifts can produce measurable changes in microbiome composition within days to weeks. Fermented foods such as yogurt, kefir, sauerkraut, and kimchi offer another route to microbiome recovery, delivering live beneficial microbes that can help rebalance the ecosystem. Reducing consumption of UPFs, especially those high in emulsifiers, artificial sweeteners, and preservatives, can reduce microbial stress and promote recovery. These small, consistent choices may help the body recalibrate, improving metabolism not through restriction, but through biological restoration. In light of these discoveries, obesity is increasingly understood not as a simple issue of willpower, but as a condition shaped by biological feedback loops in which the microbiome plays a central role. Processed foods not only add calories, They alter the very systems that govern how calories are used, how hunger is felt, and how fat is stored. Addressing obesity, therefore, requires more than counting macros or increasing exercise. It involves nurturing the body's internal ecosystem, restoring microbial health so that the body can return to its natural rhythms of satiety, energy regulation, and metabolic balance. The gut microbiome is not just a passive player in digestion, it is an active regulator of health, weight, and disease. As science continues to unravel its complex role, the message becomes clearer. A healthy gut, supported by real, unprocessed food, may be one of the most powerful allies in preventing and reversing obesity.